last in July, I spoke on another perspective of non-coding networking in plant pathogen interaction, and today I'll be focusing on the other aspect of the non-coding network. And so, just for the newcomers, we basically work on non-coding and their role in plant fungal interactions specifically, and try to find out novel non-coding molecules which could be possibly used in improvement of crop types. So yeah, so before I go deeper into my topic, I just want to focus on something specific, which is very interesting about the nature of transcription preparation. You know, all these species transcribe genes and then they encode protein. But if you see with the increase in the complexity of a genome, you will see that the percentage of the gene encoding a protein goes on decreasing. If you see a prokaryotic genome, you know, 90 to 95% of the genes encode a protein. But if you move into humans, it is almost about 3% of the total genome. So what does this mean? It means that a large amount of genes encode happens to those transcript. So it was considered to be a pervasive transcription method, a large amount of transcriptional dark matter, which is functionless, or let us say did not encode for a functional thing. But with gradual improvement in next generation sequencing strategies and genomics research, now we are very much knowing that a majority of these are the non-coding RNAs, which play significant role in altering or modulating the expression of the coding genes. And so we basically focus on those kinds of non-coding RNA and how they perform. And today, my focus is on the microRNAs. And so for that, just need to understand the coding one are those which encode tons and tons of protein to carry out functions, while the non-coding ones are those which regulate the coding of these genes, so that a complementary analysis could be created, and that would result in a balanced representation of the physiological and biochemical responses of plants to different kind of phytopathogens. And this is the classification. I don't want to go deeper into this part. Just for understanding, if we talk about the non-coding RNA, they are broadly classified into the long non-coding type and the small non-coding one, precisely based on more than length. Actually, the long non-coding one being larger than 200 nucleotides, while the smaller ones are smaller than 200 nucleotides, and they are further classified into several different types, mostly based upon their origin and sizes actually. And well, they all play somehow or rather any kind of role in the process of physiological responses to the plants. But primarily I'll be interested in talking about microRNAs today, because they are endogenous in nature and has a natural ability of having complementary binding sites with the encoded transcript inside the genome, and instead of helping them in the process, they instead of binding, they create about breakage through transcriptional inhibition or direct cleavage, and this breakage of the encoded transcript usually result in different kind of physiological responses, leading to plant growth, development, and different kind of stress responses. So we have been working on certain different plant pathosystems to understand the role of different kind of non-coding networks in all of them, precisely in a biotropic environment or in a necrotropic environment, and see what is the role of this kind of non-coding RNA in them. And before going into that, let me give you a brief idea about how exactly a microRNA work inside a plant system. It's basically encoded out of a miRNA genes or microRNA inducing genes. So a polymerase II protein result in the synthesis of a primary microRNA, and a combination of protein structures led by a complex formed by a DCL1 protein, which is a dicer-like protein, and a hyponastic lead protein and a serrate protein. This is a combination of three proteins which further process of a primary microRNA into a 200 nucleotide pre-microRNA, and subsequently the same protein complex result in the formation of a microRNA star duplex, which is almost of a size of about 20 to 24 nucleotide. Subsequently, you know, this all takes place inside the nucleus, and then there is a protein called as hasty-1 protein, which facilitates its movement into the cytoplasm. And in the cytoplasm, it comes in contact with a endonuclease protein called as Argonaute-1, resulting in the formation of a complex called as the RNA-induced silencing complex. And this complex specifically guides the microRNA, single-stranded microRNA, to the particular messenger RNA based upon its complementary site, and this binding results in cleavage of the transcript, and as such the expression of that gene is modulated. So taking this further, I'll be focusing on our discussion today on a specific pathosystem, that is Anthracnose chili pepper interaction pathosystem. So Anthracnose is a devastating disease that occurs in chili pepper, and it amounts to about 60% of the yield loss in chili pepper across Southeast Asian nation. That amounts to somewhere in the range of 4 to 5 billion US dollars. So it is indeed a very devastating disease, and it is actually caused by a species complex, not by a single species, but by a species complex. And this infects the plant by attaching to the epidermis, thereby germinating there, forming adhesive episodias, which germinates into hyphae, and that results in necrotic lesions. And this necrotic lesions results in the development of acervules, and these are the acervulocytes, which gives rise to the conidiospores. And this conidiospores subsequently again sits on the surface of a germination of epidermis of a plant, and then subsequently it again continue this cycle. And as I said, it is one of the very devastating one. The problem with the species is that there are 3 species: Coletotrichum truncatum, Gloeosporoides, and Acutatum, and they have different mode of infection, and getting a clear-cut resistance response in chili pepper is really challenging. So our lab has been working on understanding the mechanism of interaction between this interaction, and there have been certain development that has been realized in last 5 to 6 years. Several different fungicides and biocontrol agents are being used for managing the disease, but I think the most appropriate method that is available has been the host radiation breeding approach. And several R genes and quantitative trait loci has been identified with respect to resistance response against all the 3 species. However, this hasn't met with tangible amount of success, owing to the fact that the pathogen keeps on varying, and variability in the resistance response of the plant, and of course the environmental toxicity that comes up with the use of fungicide and other chemicals. So what can we think, what is beyond this? What can we think beyond our genes? So we have moved into the genomics networking and understanding how genomics can help us in getting to know about the genetic structure and the interaction between the plant and the pathogen, so that possibly we can think about a peculiar resistance response that could be built in chili pepper for executing resistance to Anthracnose pathogen. So we have carried out a couple of comparative transcriptome analysis in resistant and susceptible genotypes of capsicum annum to see what is the nature of genetic changes that is happening under a susceptible and resistance response. And not surprisingly, we have found that the variability in resistance and susceptibility to Anthracnose pathogen largely depend upon differential expression of series of genes related to MAP kinase cascade, different kind of transcription factors, then genes related to jasmonic acid and salicylic acid signaling, as well as certain genes from the NBS LLR class, as well as the PR class proteins. And also by production of glucosinolates also plays a very important role in inhibiting the growth of the pathogen in the plant system. A very interesting result what we ended up in this work is that as many as 71 differentially expressed genes were found to be targeted by microRNAs. And this clearly suggests of the fact that there is definitely a microRNA mediated silencing system that could be prevailing or playing an important role in generating resistance to Coletotrichum truncatum in chili pepper. So considering this factor into account, we designed a pipeline to identify microRNAs that would possibly induce the immunity in chili pepper against Coletotrichum truncatum. And this is the pipeline which we followed, where we selected a resistant chili pepper line, that is Punjab Lal, a widely used chili pepper line across India, and the other one being Arka Lohit. Both of them were actually developed at ICAR, and this is the basic. I don't want to go much deeper into it. As you can see, this is the normal pipeline where we go through RNA-seq and then use certain kind of tools to identify the microRNA, then subsequently the targets, and the targets are then subjected to downstream analysis. Well, this is the broad statistics, and well, I would like to focus in here. The microRNAs, what we ended up were usually of the size of 21 to 24 nucleotide, must fitting up as the requirement of a plant microRNA, and about 80% of them were intergenic in nature, and about 90 to 95% of the microRNA which ended up with a uridine nucleotide at the 5' site, clearly satisfying all the requirement for a plant microRNA. Well, the analysis resulted in the identification of about 79 conserved microRNAs in this study, as well as 23 novel microRNA, and we ended up with several of them differentially expressing, but based upon the threshold value of significant differential expression, we found that as many as 23 conserved microRNAs were differentially expressed, and about 12 of the novel RNA were significantly differentially expressed. And we particularly focused on two of them, that is MIR169A, which was significantly induced in the susceptible genotype, considering the fact that they are negative regulator, prominent negative regulator of defense, and microRNA, novel microRNA miRNA37A, which was significantly induced in the resistant genotype, considering that it is a significantly induced one and as a positive regulator. So moving ahead, we just wanted to see whether the result from the transcriptome analysis or small RNA profiling is correct or not. So we did some north blot analysis of a selected set of differentially expressed microRNA, and not surprisingly found that the data was very much similar to that. As you can see here, microRNA miRNA37A, while down-regulating in the susceptible genotype, it was significantly upregulated. You can see 3.69 fold as per not blot intensity data. It is significantly induced in the resistant genotype, and if you can see MIR169, it's going down, significantly going down in the resistant genotype. So this is a prominent negative regulator, and MIR37A is a prominent positive regulator. And so we just wanted to see, as we know, the microRNA modulate expression by performing a cleavage or transcriptional inhibition of their corresponding targets. So we also studied what is the expression pattern of the corresponding targets through qRT-PCR. And as you can see here, in most of the cases wherever the microRNA is upregulated, the corresponding target genes is downregulated. If you can see, for example, miRNA37A, the significantly induced one in the positive, the resistant cultivar, you can see here, while in the susceptible cultivar, the target is significantly going up. In the resistant cultivar, the target is significantly repressed. This suggests that there is a reciprocal alteration in the expression level of the microRNA and their corresponding targets, which clearly satisfy the fact that indeed these are some of the targets. But then, you know, always it so happens that there could be multiple targets being regulated by multiple microRNAs. So we have to be double sure whether these are indeed the actual target, actual genes that are targeted by the microRNA, or there is something else. So to further confirm this, we wanted to perform or see whether the overexpression has been realized or not. So we selected our Calo hit, which is the susceptible genotype, and developed clones to overexpress microRNA miRNA37A in those lines. So the transgenic lines were evaluated for the presence of microRNA miRNA37A. As you can see, these are the three transgenic lines selected. A heavy accumulation of the microRNA is visible, and the same can be attributed through a qRT-PCR result here, a significant upregulation and accumulation of the microRNA here. Now, if you see the target genes of the miRNA37A, they are significantly repressed. There are three target genes, three genes targeted by microRNA37A. These target genes encode for the ethylene response transcription factor, and as you can see, all the three ethylene response transcription factor were going down and down. And to further confirm it, the pathogen infection assay clearly showed that while the empty vector 1, which was not having miRNA-37A, showed a significant symptomatic development of the pathogen, the 3 transnuclein overexpressing microRNA miRNA-37A did not show any kind of symptomatic development, or the symptoms were highly negligible. And this can be further corroborated with the point that there was significant decrease in the sporulation and the relative fungal biomass in the transnucleins overexpressing miRNA-37A, clearly suggestive of the fact that if we overexpress microRNA miRNA-37A, it is possible to enhance resistance to Anthracnose pathogen. And to further confirm, we wanted to see whether the targets were actually cleaved or not. So we performed RLM Race, the rapid amplification of cDNA, which is a gold standard used for understanding finding out the cleavage sites of the microRNA target genes. And so, as you can see, these are the 3 targeted ethylene response factor, and the microRNA could perfectly cleave at a particular point about 11 nucleotide from the 5' end, and perfectly in all the 3, clearly suggesting that these are indeed the possible targets of the microRNA. So inhibition of this target possibly facilitate enhanced resistance to the pathogen. And again, you need to again double sure whether the transcriptional inhibition of the target gene is actually facilitating the resistance response or not. So we wanted to see whether the microRNA is actually inhibiting the effect or cleaving the action of the target gene at the transcriptional level or at the translational level, because microRNA can behave at different levels, So we wanted to study that. So a co-expression analysis experiment was performed in Nicotiana plant, where we designed vectors having the target gene and certain vectors having both the target genes as well as the microRNA, and a third vector having the target gene plus a different microRNA which do not have a cleavage site with the specific target gene. And as you can see here, the transnucleins with a distinct construct, whereas if you see the one which is having only the gene was properly induced, and you can see that the one where both the microRNA and the target genes was there, it was significantly repressed. And the one where a different kind of microRNA, which is possibly not a particular targeting microRNA for the gene, did not show much of a change, so suggesting the fact that microRNA miRNA-37A indeed cleaves the target gene at the appropriate point. And this was further validated by Western blot result here, where we designed flag tag antibodies of the microRNA as well as the target. And you can see here, in presence of microRNA, there is no accumulation of the target proteins, or a very negligible accumulation of the target protein in all the 3. So clearly suggestive of the fact that microRNA miRNA-37A indeed targets the ethylene response factor and inhibit them at the transcriptional level to enhance Anthracnose resistance in chili pepper. So this makes us clear that the inhibition of the ethylene response factor, or the silencing of the ethylene response factor, is possibly one of the reasons that is facilitating the enhancement of disease resistance to Anthracnose pathogen. So again, we wanted to ensure whether this is true or something else, other genes are involved in the process. So to ensure that we perform a virus-induced gene silencing system, where we try to silence the ethylene response factor genes in the overexpressed lines of alkaloid, and to see whether it is bringing about any kind of changes in the resistance, in the infection level of the pathogen. As you can see here, this is the wild type alkaloid, which is a susceptible genotype with heavy infection, and this is a VIX lines with the empty TRV vector, still a lot of infection, and this is the resistant genotype, no silencing of the gene, and there is no infection. no infection here. It means that here I'm having a excessive production of the microRNA, and subsequently the target is already in negligible amount, whereas in this one the target is very high, and so the overexpression of the target is actually facilitating the infection to occur. So what we did is we silenced these alkaloid lines for the particular ethylene response factor. These are the 3 ethylene response factor silence lines, as you can see here. And you can see when we silenced the target genes, there was no pathogenic infection in any of them. So this clearly is suggestive of the fact that overexpression of the ethylene response factor genes actually facilitate infection by choledotrichum truncatum, while overexpression of microRNA miRNA37A inhibit the target protein and thereby bring about a resistance response. So what we conclude here is that microRNA miRNA37A is a major determinant in paper immunity, and simultaneously I would like to say that the target ethylene response factor is a prominent determinant that facilitate pathogenicity in Anthracnose pathogenicity in capsicum annum, and possibly we can bring about resistance response by bringing about RNAi or CRISPR-Cas mediated genome editing. And we are actually at an advanced stage of silencing ethylene response factor genes in certain anthracnose susceptible variety, so as to generate resistance response in them. We have some leading results on that. Possibly we can discuss it in some other presentation. And so what are the future leads in here? So the identification of this kind of small RNA factors can play a very important role in shutting down plant defense to choledotrichum truncatum. And then here miRNA37A is a major determinant for paper immunity against choledotrichum truncatum. Mind you, I told you in the beginning there are 3 different choledotrichum species which are responsible for the anthracnose infection. So we need to see whether miRNA37A also have a similar kind of resistance response, and the work is still in progress. And we are also looking into the other part where if there is any kind of pathogenic small RNA which could be acting as an effector in a cross-kingdom RNA interference process. And there have been evidence in the recent times that some kind of, apart from a protein effector, some pathogen also produces small RNA which can bring about suppression of the immunity. So apart from the ethylene response transcription factor, we are also looking into certain kind of pathogenic small RNA which can be identified, and they could be possibly suppressed to induce defense response in chili pepper. And of course, we are also trying to develop them into markers so that a quick identification of the disease can be done, and that is a priority in the breeding program. And with this, this is my group, and I like to thank NatCap Professor Nat is from Canada, and he supports the long non-coding work. And Professor Kai Jun is from Chinese Academy of Agricultural Sciences, and he is helping us with our CRISPR-Cas genome editing work. We are still continuing with that one, and leads are coming in, so it is still at its infancy. And this is my group. Rukmini has done her PhD in microRNA, and most of the molecular works he has done, and Satya and other guys have also done certain work